hydrogen ions in a solution, meaning that a lower pH indicates a higher concentration of the hydrogen ions. This is because the formula used to calculate pH approximates the negative of the base 10 logarithm of the molar concentration of hydrogen ions in the solution. More precisely, pH is the negative of the base 10 logarithm of the activity of the hydrogen ions. So pH is abbreviated as power of hydrogen or potential for hydrogen, and is a scale used to specify how acidic or basic a water-based solution is. And it is defined as negative log of the hydrogen ion concentration and is inversely proportional to the hydrogen ion concentration, expressed as pH minus log hydrogen ions. Okay, so I know you must all be aware of the pH, because you must have come across this topic, and you all must have a very good knowledge of pH. So it won't be difficult for you to understand the lecture. So the acidic and basic solutions, as you all know, acidic solution is a solution the pH of which is less than 7. So it has a higher concentration of hydrogen ions. Basic solutions, on the other hand, have a pH value above 4 and above 7, and they have a very low concentration of hydrogen ions. So pH value of some important biological fluids, intestinal juice has around the pH of 7 to 8, blood has 7.35 to 7.45, gastric juice is 1 to 2, urine has 6 to 6.8, and saliva has a pH of 6.6 to 6.8. So basically there are three methods of how you determine the pH of a solution. So these three methods are by using an indicator, use of a litmus paper, and by using pH meter. pH meter is the most precise and accurate method. So let's discuss how you're gonna determine the pH of a solution by using litmus paper. So basically, litmus is a water-based mixture of different dyes extracted from lichens. It is often adsorbed onto the filter paper to produce one of the oldest forms of pH indicator, and it is used to test materials for either acidity or basicity. So litmus paper is a filter paper which has been treated with a natural water-based soluble dye obtained from lichens. The main use of litmus is to test whether a solution is acidic or basic. Litmus paper comes in blue color, red color, and purple color where it is a neutral litmus paper. So the object of this practical is to determine the pH of a given solution A and B by using litmus paper. So the principle on which litmus paper works is the red litmus paper contains a weak diprotic acid, meaning it has two hydrogen ions. So when it is exposed to a basic compound, the hydrogen ions react with the added base. The conjugate base formed from the litmus acid has a blue color, so the red litmus paper turns blue in alkaline solution. So when the litmus paper, red litmus, reacts with a base, when it is dipped into a base, it releases hydrogen ions. These hydrogen ions react with a base to form a conjugate base, and that conjugate base gives blue color to that red litmus paper. On the other hand, when we use a blue litmus paper, and then when we put that blue litmus paper in the red litmus paper, or in an acidic solution, it turns to red by the same mechanism, of course. So blue litmus paper basically contains a conjugate base, and that base reacts with the acid to give it a red color. So this is all about the principle. Let's move on towards the procedure. So um, although you're not going to perform this practical, unfortunately, but this is how we do it in the laboratories. So you're gonna take two beakers and you're gonna mark them A and B. So in beaker A, you'll add 5 milliliters of a given solution A, and 5 milliliter of given solution B will be added in the beaker B. So you will add both red and blue litmus papers in both the beakers, and you will see the color change. So in beaker A, we usually give you an acidic solution. So the blue litmus paper will turn to red, and there will be no change in the color of red litmus paper. While in beaker B, the red litmus paper will turn to blue, and there will be no change in the blue litmus paper. So beaker A contains an acidic solution, and beaker B contains an alkaline solution. So there are certain limitations to the use of litmus paper. First of all, it cannot yield a numerical pH value. It just gives you a rough estimate of what could be a possible solution which is present, which is given to you. So it does not yield a numerical or the exact pH value. Also, it can change color for other reasons besides an acid-base reaction. So these are two big limitations to the use of litmus paper. This is how you perform the practical. These are two big beakers, and blue litmus paper turns to red in an acidic solution. Red litmus paper turns to blue in a basic solution. This is a universal pH paper. We will discuss it later on. So now the determination of pH by using indicator. So pH indicators are halochromic chemical compounds which are added in small amounts to the solution so that their pH can be determined. So a pH indicator detects the hydrogen ions or hydronium ions basically. So the principle on which these pH indicators work on, it's almost similar to that of the litmus paper. So pH indicators are weak acids or weak bases that exist as natural dyes and indicate the concentration of hydrogen ions or hydronium ions in a solution via color change. Indicators are various organic dyes of natural and synthetic origin. So they change color as pH of solution is changed. So this is the principle that pH indicators detect the presence of hydrogen or the hydronium ions, and they do this by reacting with hydrogen ions and the hydroxide ions. So they're themselves weak acids or bases. So if an indicator is a weak acid, its conjugate base has a different color, and vice versa for the weak bases. So this is how a pH indicator works. So there are some pH indicators we commonly use in a laboratory setup. So thymol blue, methyl blue, phenol red, and phenolphthalein. These are four major indicators that are commonly used. These are their pH ranges, and these are so methyl blue and so methyl red and thymol blue are towards the acidic side, and phenol red and phenolphthalein are basic. So thymol blue will give you a red color when it is when there is an acidic solution, and will give you a yellow color when there is an alkaline solution. Methyl red will give you a red color in an acidic solution and a yellow color in for an alkaline solution. Phenol red gives yellow color in an acidic medium, while it gives a red color for the alkaline medium. And the phenolphthalein will give you no color, or it will be colorless in an acidic solution, while it will give a purple pink color in an alkaline solution. So the object again is to determine the pH by using an indicator. So the reagent we are going to use here is the methyl red indicator. So you will take two test tubes and you will mark them A and B. In test tube A, add 5 ml of given solution A and a drop of methyl red indicator. In test tube B, you're gonna add 5 milliliter of given solution B and a drop of methyl red. So generally we give test tube we give you an alkaline solution in test tube A and an acidic solution in test tube B. So again, you're not going to perform these practicals in your examination, so don't worry about it. This is just to explain how you perform practical in your lab. We will not ask you to perform these in your examination. So yellow color is produced because there was an alkaline solution in test tube A, and red color is produced due to presence of acidic solution in test tube B. So this is methyl red indicator and this is phenol red. There are certain limitations to the use of pH indicator. Again, pH indicator measure pH at a very low accuracy. It does not yield results in a numerical value, so it cannot give you accurate results, right? Also, for this test, for the use of any indicator, it is necessary that the solution you want to test should be colorless, because if there is any colored solution, how will you notice the change, or how will you observe the change? So the color of the solution there should be colorless solution for the test to be positive. So you have to use a colorless solution if you're using an indicator. So this is the universal indicator. So universal indicator basically is a blend of pH indicator solutions. As we discussed, there are 4 or 5 different types of pH indicator solutions. So universal indicator is a blend of those pH indicators, right? So and this universal indicator basically is designed to identify the pH of the solution over a wide range of values. So it can give you a very wide range of results. It is not confined to just produce a color, but it will give you some sort of numerical value also. So there are several different formulas for universal indicators, but most are based on the mixture of thymol blue, methyl red, bromothymol blue, and phenolphthalein. So these 4 are combined together and they form a universal indicator. So for composition of universal indicator, apart from these four indicators, water and propane 1-ol and sodium hydroxide are also needed. So water and propane 1-ol are used as solvent because they both are polar and dissolve all the other ingredients in the solution. So water and propane 1-ol are used as solvents. Sodium hydroxide is an alkali, and it is used to provide an alkaline medium to the solution, so that the pH adjusts and to ensure that each color is shown at a correct pH value. It is necessary to add NaOH because some of the indicators, for example methyl red, is acidic, and that's why, and to neutralize the effect of that acid, we have to use an alkali. So yeah, that's the reason we use the sodium hydroxide. So types of universal indicator, there are 2 main types of universal indicator. One is in the paper form and others are solutions, as we discussed. So paper form is a strip of colored paper which changes color to red if the solution is acidic, and to blue if the solution is basic. The strip can be placed directly onto the surface of a wet substance, or few drops of solution can be dropped onto the universal indicator. Okay, so the color that appears on the paper form can then be compared to a chart which is provided here. As you can see here, this is the universal indicator paper. This is the paper. You're gonna dip this paper into a solution, and the color that appears on this paper, you will match this color with this um, with this with these values, okay, with these colors. So this is how pH paper gives you an estimate or an idea of of what is the pH of the solution. So it is again a better method rather than using the litmus paper and the indicators alone. So this was all about the paper form of universal indicator. Now coming towards the solution, which is a mixture of again four indicators, and this is beneficial. This will be beneficial if you have a colorless solution. So yeah, that was all about the paper form and the solution form of the universal indicators. So now coming towards the detection or determination of pH by using the pH method. Meter, this is the most precise and most accurate method of pH determination. So pH meter is pH meter basically measures the solution pH by measuring the electrical potential differences between the pH electrode and a reference electrode. We will discuss these electrodes later. So pH meter is a scientific instrument that measures the hydrogen ion activity in a water-based solution, indicating its acidity or basicity, which is expressed as pH. Okay, so pH meter measures the difference in the electrical potential between a pH electrode and a reference electrode, and so the pH meter is sometimes referred to as potentiometric pH meter. So basically a pH meter detects the pH by measuring the potential difference between the two electrodes. Okay, there are three types of pH meters. These are handheld portable pH meter, benchtop, and continuous inline pH meter. So yeah. So the parts of pH meter comprise basically of a voltmeter, which is attached to a pH responsive electrode and a reference electrode. So the pH responsive electrode is usually a glass electrode, and the reference electrode we use is usually a mercury or mercurous chloride electrode. When the two electrodes are immersed in a solution, they act as a battery. So the glass electrode, this is the glass electrode. It develops a certain electrical charge or electrical potential, and this electrical potential generated by the glass electrode is related to the hydrogen ion activity in the solution, and this hydrogen ion activity in the solution is measured by the voltmeter here, and measured. This hydrogen ion activity basically is related to the potential generated by the two electrodes, and then this electrical potential is measured by the voltmeter. So the electrical current generation is related to the hydrogen ion concentration. So this is the principle by which a pH meter works. I would like to read the slide for you again, that a pH meter basically comprises of a simple electronic amplifier and a pair of electrodes, or alternatively a combination of electrodes, and some form of display calibrated in pH units. It usually has a glass electrode and a reference electrode, or a combination electrode. So basically there are some types of pH meter which have separate glass electrode and reference electrodes, but nowadays combination electrodes are used, in which there is a single electrode and both the glass electrode and the reference electrode are merged into that single combination electrode. So these are the parts of pH meter, and the principle is pH meter measures the voltage between the two electrodes and display the result converted into corresponding pH value. On immersion of the glass electrode and the reference electrode in the test solution, an electrical circuit is completed in which there is potential difference created and detected by the voltmeter. So the electrical current generated is related to the hydrogen ion concentration. So I hope I'm clear up till now. Let's move on towards the procedure. Again, we used to perform this practical in our lab, and we take two beakers here again, beaker A and beaker B, and we put our desired solutions in both the beakers, and then we put pH meter for 30 seconds in both beakers, and then we note the readings. So for pH meter, calibration is very crucial. Calibration means to mark a gauge or an instrument with its standard reading. Okay, in other words, it is to correlate the readings of an instrument with those of the standard in order to check the accuracy of the instrument. So calibration is important because if your pH meter is not working properly or is not calibrated properly, it will give you wrong results. So when you see a pH meter says, for example, pH 7, you have to make sure that the pH is actually 7. So for that, you have to calibrate your pH meter before taking the readings. So how a pH meter is calibrated? Buffer solutions are used to calibrate pH meters. Why? Because they resist any change in the pH. Okay? So calibration is done by using measuring substances with known pH levels and setting the pH measurements to those levels on the pH meter. So for calibration, we take buffer solutions of known pH levels, and we put our pH meter in those solutions and calibrate our pH meter accordingly. So calibration is first done with a buffer of pH 7. Then if you want to check or test or estimate the pH of an acidic solution, you put your pH meter in an acidic solution of pH 4. Or if you want to check a basic solution, then you first calibrate your pH meter into a buffer solution of pH 10. After that, you are going to perform your test. So this is how calibration is done, and this is the importance of why we calibrate. This is really important. So now the significance of pH. So pH is important in vast areas of life, from our body to the environment, to the animals, to the plants. Everything is working on an optimum pH, right? So pH is important, and it is a quantity basically that reflects the chemical conditions of the solution. So we are surrounded by solutions, okay? So it can control the availability of nutrients, it can control the biological functions, microbial activity, and behavior of chemicals. So pH significance of pH is important, or pH is significant, there's a lot of significance of pH in the human body, in agriculture, in the water treatment, in food industry. Everywhere we are surrounded by aqueous solutions and their pH. So So, yeah. Um, so, optimum functioning of the human body. All processes occurring in the body need a balanced pH to work efficiently, right? Acid-base balance is essential for the best functioning of the body. So, acid-base balance is really important for the functioning of human body, from the intake of the food through the mouth until its excretion. Everything is pH dependent, right? uh, Salivary amylase, it works on an optimum pH. The digestive enzymes, they work on an optimum pH, and the cells work on an optimum pH. So nothing can be done without an optimum pH, and uh, life cannot work, or life cannot proceed without an optimum pH. So significance of pH cannot be denied. It is also very much essential in the field of agriculture and gardening, because soil is a complex system that involves many different factors that are affected by soil pH, such as microbial activity, fungal growth, availability of nutrients, etc. They all depend on a particular pH. Okay. In terms of aquaculture and aquatic ecosystems, so water that has a pH that is too low or too high can be harmful to the fish and other aquatic life. So again, pH is crucial for the aquatic ecosystems too. For the water, whether it is drinking water or wastewater, pH is important. The pH of drinking water should be between 6.5 to 8.5. In food industry, pH is measured to test for the quality, to control microbial activity, to control the taste and other properties, and to prolong the shelf life of the food. So this is the significance of pH. Now we're coming towards the buffers. So what is buffer? Buffer is a solution containing weak acid-base pair that is able to accommodate the addition of strong acids or bases without the pH of the solution changing much, right? So buffer usually is added when we do not want to change the pH of our system. Buffering is important in the living systems as a means of maintaining a fairly constant internal environment, which is also known as homeostasis. So a buffer is a chemical substance that helps maintain a relatively constant pH in a solution, and it is defined as a solution which resists the change in the pH when a strong acid or base is added in a small amounts. Chemically, they are made up of weak acid with their conjugate base, or vice versa, in a fixed ratio. So buffer systems of the body. So there are four basic buffer systems in a human body. These are the bicarbonate, or the phosphate, hemoglobin buffer, and the protein buffer. Bicarbonate buffer consists of carbonic acid and bicarbonate ions. They help regulate the pH of blood. So when the blood pH drops into the acidic range, this buffer, the bicarbonate buffer, acts to form carbon dioxide gas. This gas is then expelled by the lungs out of our body during the process of respiration. So, and during alkaline conditions, this buffer brings back pH to the neutral levels by causing excretion of the bicarbonate ions through the urine. So this is how the bicarbonate buffer acts. The phosphate buffer is composed of hydrogen phosphate ions and the dihydrogen phosphate ions. So on conditions when there is an excess of hydrogen ions, or when there is acidity, the hydrogen or the excess hydrogen enters the cells. It reacts with the hydrogen phosphate ions, which accepts them. So the acidity is normalized. And during alkaline conditions, the dihydrogen phosphate ions accept the excess hydroxide ions that enter the cell. So this is how a phosphate buffer works. Hemoglobin buffer has an ability to bind with either protons or oxygen at a given point of time. So basically what happens here is binding of one releases the other. So what happens in a hemoglobin buffer, that in hemoglobin the binding of protons occur in the globin portion, whereas oxygen binds at the iron of the heme portion, right? So if you're exercising, the protons are generated in excess, right? Hemoglobin helps in buffering by binding these protons and simultaneously releasing molecular oxygen. So binding of one releases the other, right? So this was the hemoglobin buffer, and now coming towards the protein buffer. Proteins consist of amino acids held together by peptide bonds. The amino acid possess an amino group and a carboxylic acid group. And as we discussed while we were detecting the proteins, or while we were detecting the quality of the proteins, we discussed the nature of amino acid, that they act as switcher ion. They act as switcher ion at a physiological pH. So at a physiological pH, carboxylic acid exists as carboxylate ion with a negative charge, and the amino group exists as NH3 plus ion. So when the pH becomes acidic, the carboxyl group takes up excess hydrogen ions to return back to the carboxylic acid form. If the blood pH becomes alkaline, there is a release of protons from the NH3 plus ions, which takes the NH2 form. So this is how a protein buffer acts. So I hope we are clear up till now. Let's move on towards the significance of Henderson-Hasselbalch's equation. So basically, this equation helps to estimate the pH of a buffer solution and gives the ratio of the conjugate base to the conjugate acid. It gives a quantitative correlation between pH, pKa, and the buffering capacity, right? So this is the formula for of the Henderson-Hasselbalch equation. pH is the acidity of the buffer. Ka is the dissociation constant of an acid. This is a constant value. pKa is the negative logarithm of the Ka, or the dissociation constant of acid. A is the concentration of conjugate base, and Ha is the concentration of conjugate acid. So if the base and acid are equal in concentration, pH will be equal to the pKa, or the dissociation constant, right? So this was all about the Henderson-Hasselbalch equation. So now we're going to perform a little exercise here. We're going to determine the pH of a buffer solution using the Henderson-Hasselbalch equation. So the formula for the pH, the formula for this is the pH of buffer solution can be calculated by pH is equal to pKa plus log base upon acid. So you will going to perform a little exercise here, and the reagents we are going to be using here is 0.2 m acetic acid, 0.2 m sodium acetate, and a pH meter. So for acetic acid, the pKa is 4.7. It's a constant value. Okay. So again, you're going to take 3 beakers, and first we will make buffers. Okay. So in beaker A, you will take 2 mils of acetic acid and 20 mils of sodium acetate. In beaker B, you will take 20 milliliters of acetic acid and 20 milliliters of sodium acetate. In beaker C, you will take 20 milliliters of acetic acid and 2 ml of sodium acetate. Mix all the contents, and then we will know the pH. So basically, we have formed 3 buffers here, and then we will measure their pH. Okay. So coming towards the calculation, For example, for beaker B, since we take equal amounts or equal concentrations of acetic acid and sodium acetate, so there is an equal ratio of these both. So for so formula for this would be pH is equal to Pa plus log base upon acid. So base and acid were both in an equal ratio, so we're going to put 1 over 1 here, and log 1 is equal to 0. So in this case, when there are equal amounts of acid and base, the pH will be equal to the pKa, right? So as we know that the pKa for this was 4.7, so meaning that the pH will also be 4.7, right? So then we will put our pH meter in the solution, and then we will check our results. Okay, so for beaker A, the pH would be high because there was more sodium acetate, so there will be high pH. In beaker B, as we already calculated, pH will be equal to pKa, which would be 4.7. And beaker C, pH would be low than pKa because more amount of acetic acid was added in the beaker. So this was all about the calculation. So if you want to calculate the pH for beaker A and pH for beaker C, you're more than welcome. So now the standard operating procedure, or the SOP of pH meter. Well, this is just to give you an idea of how a pH meter works. Okay, so you do not have to learn it step by step, and you do not have to remember the exact temperatures or the amounts which are used in this. But just to give you an idea of how a pH meter works, you should go through this SOP. This is the SOP. Take some time to read it out, and yeah. So again, I would like to tell you about the calibration. We have made 4 buffers here, one of pH 4, one of pH 7, and one of pH 10. And before taking our readings of the given solutions, we always calibrate our pH meter. If we're going to test for an acid, we will first calibrate our pH meter with a buffer of pH 7, and then with the buffer of pH 4. If we're testing for an alkaline solution, we will again place our pH meter in a buffer of pH 7, and then in a buffer of pH 10. So this was all about